The process of protein synthesis occurs in the cell. DNA exists in the nucleus, which contains all the genetic information of an organism. Chromosomes are found in the nucleus. They are thread-like structures made up of DNA that is tightly coiled around histones, a protein. A strand of DNA is composed of nucleotides, which are the monomers of DNA. A sequence of nucleotides is known as a gene. The process of protein synthesis begins with RNA polymerase. The RNA polymerase binds to a specific region on the DNA, unwinding and separating the strands. As the RNA polymerase moves along the DNA, it reads the DNA nucleotides and matches each nucleotide with a free-floating RNA nucleotide. This synthesizes a new strand of nucleotides, known as messenger RNA, or mRNA for short. While the mRNA strand is now complete, some sections of nucleotides will not code for proteins. These sections, known as introns, are removed from the mRNA in a process called splicing. After introns are removed, the nucleotides that code for proteins, known as exons, are rejoined. The mRNA is now mature and ready to exit the nucleus into the cytoplasm, ending the first part of protein synthesis, which is transcription. The mRNA drifts into the cytoplasm and encounters a ribosome, which begins the second part of the process known as translation. Translation involves the conversion of the mRNA sequence into a sequence of amino acids, which forms a protein. These amino acids are attached to transfer RNA molecules, which are free-floating in the cytoplasm. Each amino acid is coded by a different sequence of 3 bases, known as anticodons. The ribosome reads the mRNA sequence 3 bases at a time, known as codons. It matches the complementary anticodon on a tRNA molecule with the codon on the mRNA, and transfers the amino acid attached to the tRNA to the growing polypeptide chain. This is a chain of amino acids joined with peptide bonds, which goes on to fold into a protein.